Okay. Well, hello, everyone. Thank you all for attending this webinar. My name is SeungOok, and today I'll be introducing the whole exome sequencing solutions provided by Vazyme. And this webinar is meant to be an introduction to whole exome sequencing related to products. So from now on, I will refer to the whole exome sequencing as WES, W-E-S, WES. And I hope you find this session informative and that it answers your questions. So before we get into the WES part, I'd like to introduce our company a little bit for those who are not familiar with Vazyme. So Vazyme is a global technology company dedicated to the life science. And year 2012, our journey started with the development of genetically engineered recombinant proteins slash enzymes. Soon, our business expanded into various segments, such as in vitro diagnostics, next generation sequencings, nucleic acid extractions, um, and etc. So currently, we extend our service to clients in more than 60 different countries and regions with the subsidiary companies located in the US, Germany, Korea, Singapore, UK, Canada, Hungary, and so forth. So our company was enlisted in Shanghai Exchange um, Stock Exchange in year 2021, and our revenue exceeded 500 million US dollars in 2022, and we continue to grow. So our mission is simple, and it is to empower our customers to advance the innovation of science and health. And we are driven by a singular mission, but we address multiple responsibilities to fulfill it. So first, reproducibility. We ensure the lot-to-lot consistency for all applications. Performance, we strive to provide the best products. Convenience, we anticipate efficiency and accuracy from our users through our innovation in technology. Equipment, we supply necessary products that are needed for your research. Last but not least, service, we prioritize your satisfaction. So not to mention, we collaborate with both academic and industrial researchers. We have more than 60,000 publications so far, and we strive to develop innovative products. And thank you so much for all those who believe in us and trust our products. We also want to mention that our company is furnished with a strong supply chain for reagents that can produce 50 million reactions a day. And we are committed to comply with international standards. As you can identify all of these certificates for medical devices, general quality management, and safety and health management, and environmental management systems. So today's topic has the following agenda. I will briefly touch on the WES background and our WES solutions. Specifically, we will emphasize the power of automation that can bring. And then I will share some of the results. And also, I want to talk about our previous experiences with our customers. And that will conclude our meeting. So what is WES?
So it is next generation sequencing method or technology that involves sequencing the protein coding regions of the genome. So like the pictures on the left, the genomic DNA consists of exon and intron. An exon, which is the protein coding region, and intron is non-coding region. And when it comes to the ratio of a protein coding region is about 1%. And this is significantly small amount, but it contains majority of the known disease-related variants, like up to 85%. And WES helps identify disease-causing mutations, discover new disease genes, and it can also guide targeted therapies, and it can enhance our understanding of genetic variations, evolutions, and disease susceptibility across the populations. So typically when you compare the whole exome sequencing to whole genome sequencing, the WES is targeting gene coding sequence, whereas WGS is covering the whole genome. So essentially what you get out of the both technology is about pretty much the similar, but WGS contains more information that WES cannot provide because it can cover non-protein coding regions as well. So it is more costly, but when you compare the sequencing depth, the WGS requires a lower sequencing depth um, to provide a higher coverage at a lower cost. So whole exome sequencing is a next generation sequencing technology that is getting more popular, as you can see from the market trend graph. So the market expansion was only slowed down during the COVID-19 pandemic when the R&D funding was reduced and global lockdown occurred. But the global whole exome sequencing market um, was estimated at USD of $472.9 million in 2024, and it is expected to grow at a CAGR of 18.4% from 2025 to 2030. And in recent years, the market demonstrated a huge expansion potential due to the new technologies developed, and there is growing need for accurate and early detections of genetic and chronic diseases. And when you look at the Europe and Asia Pacific regions, the situation is pretty much similar. In Europe, um, the market size is estimated to be a little over 120 million US dollars, and it is expected to grow at a CAGR of 17.2% from 2025 to 2030. In Asia Pacific, the market size is estimated at almost 90 million US dollars, and it is expected to grow at a CAGR of 21.6% from 2025 to 2030. So the demand for the WES is growing, and revenue is expected to rise due to increasing use in the diagnostic applications. For example, the COVID-19 highlighted the importance of the accurate diagnostics and further driving interest in OS. And medical professionals and researchers use this method to identify genetic diseases and mutations linked to the tumor growth, for example. And additionally, the advancements in the technology, automations, and data analysis tools and other technologies are reducing the cost of a whole exome sequencing. And there is a growing need for personalized care, which is another key factor. as WES enables doctors to tailor treatments based on a patient's unique genetic profile and disease risk. So I gather some of the examples where WES is applied. So first, the top left corner, it is about coloboma and microphthalmia, which is a congenital eye malformations, which is a birth defect. And when baby is born with this, and he or she will have visual impairment. So for a long time, scientists were having a tough time to identify mutations on which genes had to do with these diseases. And so using WES, they identified abnormal mutations on 7 different genes that lead to the coloboma and microphthalmia. And top right corner is the familiar hypercholesterolemia, and which causes the elevated low density lipoprotein, which is LDL cholesterol level, and that increased risk of premature myocardial infarction. So in this paper, the WES screening against the patients with coronary artery disease indicated that patients has a familiar hypercholesterolemia. And so WES was used as a diagnostic tool. And on the bottom, WES is considered as a gold standard for TMB, which stands for tumor mutational burden. And experts recommend using WES to assess TMB for it is comprehensive coverage and accurate mutation quantifications and for unbiased mutation detections. And some new research has shown that WES is very helpful in terms of finding more diagnoses than using conventional methods like chromosomal microarray assays and fluorescence in situ hybridizations. So WES plays an important role in terms of identifying the tumor variants to monitor MRD, stands for minimal residual disease, and recurrence. So researchers set a panel of, in this case, researchers set a panel of 21 common and stable single nucleotide polymorphisms, acting as a reference point for comparison. So for normalization of the data. And so before cancer treatment, tumor samples such as like FFPE are collected from each patient and undergo deep sequencing to profile personalized mutations. And then they selected a personalized ctDNA tracking site. And it was added to the panel. So this created an individually customized MRD monitoring panel. So later, when a liquid biopsy sample was analyzed to detect MRD, so in this way, the WES helps identify patient-specific MRD, improving post-treatment monitoring and personalized care. So although we have witnessed the power and the benefits of the WES, still we are facing more challenges, like such as complex data, long processing times, and lack of infrastructures in many clinics, and the need for highly skilled personnel with ongoing training. So here are some what we can offer to you for your WES experiment. So this is basically the workflow of the WES, and the red check mark is what we can provide. So from GDNA extractions and library preparations, and hybridization reagents to sequencing and software analysis. We can provide all the reagents and equipment except the sample preparations and the sequencing modules. So today I'd like to highlight a couple of automatic procedures that our company can provide with. So for the extraction step, genomic DNA can be obtained from peripheral blood, swabs, or FFP samples. And we can offer two types that allow you to choose between the manual and automated solutions based on your needs. So for automated extractions, so we provide two flexible options with the different high throughput capacities. So VNP32, which is suitable for processing up to 32 samples, and it offers flexibilities for smaller workflows. Whereas VNP96, this one is ideal for labs that handle large volumes of nucleic acid extractions. And additionally, we offer specialized reagents tailored for different sample types, including FFPE, blood, and swabs, that ensures efficient and reliable extractions. And these automated nucleic acid extraction takes between 32 minutes to 52 minutes. And another automated procedure is automatic workstation. Both library preparations and hybridization capture process can be loaded in this instrument. So for the library preparation step, we offer two fragmentation method, mechanical fragmentation and enzymatic fragmentations. And sample types that have been tested were peripheral blood, FFPE, swabs, and amniotic fluid. And based on sample types or DNA input conditions, you may choose the right method for your research. But let's say that if you're handling the cfDNA, for example, cfDNA is already short enough and with average size of 150 base pair, so it does not require fragmentations. So whereas if it is genomic DNA, the fragmentation will be required. So since mechanical fragmentation method has fragmentation process separate from the end repair and detailing, so you can skip the fragmentation and process with end repair and detailing. So however, the enzymatic fragmentation has all three reactions, which is the fragmentation plus end repair plus detailing. So genomic DNA will be suitable for enzymatic fragmentation instead. So once you are completed with the DNA library amplifications, and soon you will do the hybridization capture process. And we have COT1 DNA and blockers to increase the specific binding of the probes, thus improve the capture efficiencies. And it is biotinylated probes that target genes and later can be isolated via the beads that is appended with strep avidin. So, and for the target capture process, that involves library samples to be pulled and to be concentrated. And we offer two different options for library concentrations before the hybridization capture steps. One is magnetic bead concentration, and the other is vacuum concentrations. So when you're handling the vacuum concentration method, it uses a vacuum concentrator to concentrate the library. And its operation time varies, depends on the machines. So typically it takes about 30 minutes to 2 hours, and it provides a slightly higher sequencing the uniformity. Whereas if you are doing the magnetic bead concentrations, for example, then um, it eliminates a need for a vacuum concentrator. So that reduces lab investment. And it is easier to implement for new labs. And the operation time is about 30 minutes. So yeah, you can choose either or. So for library preparation and probe hybridization process, there are no better dark night than this automatic liquid handler, also known as VNL96. So this instrument has the in-HECO PCR module for library amplification, probe hybridization, um, temperature-sensitive incubations, etc. And there is a one temperature-controlled thermal shaker. And so this is for the magnetic beads preparations and sample and reagent mixings, etc. And he's not showing in this slide, but there are two additional temperature control modules, and there's a one magnetic frame that's for the purification purpose. And we have one 96 channel pipette, which is flexible between 1 and 96 pipette. And one robotic gripper is located right next to each other. And this can move either the plate or the lead freely around to facilitate the automatic process. And we also have 24 plate positions that are stackable so that it minimizes human labor. So with the Vienna 96, you would expect 3 to 4 hours for typical DNA library preparation for 96 samples. And when it comes to the WES, you would need at least 25 hours to get single sample done manually, whereas 96 samples can be done in the same amount of time with the automated liquid handler. So a couple more features will provide convenience to your laboratory experiences. So with our VNL96, you don't need to study programming languages like Python or Java. Understanding logical flow suffices the entire automation. And also the number of the tip usage is highly flexible. So you can choose between 1 and 96. And these features not only saves your consumables, but also enhances the efficiency of library preparations and hybridization capture performances. And we also have library preparation reagents already prepared in a plate. So following 4 points can add to your considerations for optimizing the experiments. It's ready to use, so no manual aliquoting, so that reduces errors and that ensures consistencies. It can boost the efficiencies. It can cut down processing time and speed up the library preparations. And also automation is optimized, so fully compatible with Vienna 96 for seamless integrations. And it's reliable and consistent, which means it minimizes the batch-to-batch variations for high quality data. So let me share some of the data with some metrics that you will need for quality controls. So these are performance comparison between Vazyme's products and other commercially available products, which is done internally. And the sample was the reference gDNA, and reads on target rate. So basically, Vazyme products show the highest reads on the target rate, and basis on the target rate, Vazyme's the percentage of the total sequenced bases that align to the intended target region is among the highest. And when you look at the fold 80 data, the lower fold 80 value means it requires a fewer regions to be sequenced to achieve sufficient depth across the entire target regions. So with low fold 80, Vazyme was able to achieve uniform coverage and also target coverage. The number of times that a base in the target region is sequenced and higher coverage is better. And Vazyme showed the both in both cases the highest coverage. And the duplication rate, you don't want to have high in the duplication rate. And it won't be entirely covering because it won't be entirely covering the target regions. And it is the lowest out of all. And so this indicates that Vazyme's WES performance quality is superior than some of the commercially available products. And as I mentioned earlier about our automatic solution for WES preparation, you may wonder how automatically prepared samples are different from manually prepared samples. Or how close are they? And when you compare the performance between the manually done and automatically perform the data, and you won't find a significantly different data quality. And this is because our automatic liquid handler engineers are highly trained to set up the instruments for optimal performance. So it's like having an expert performing the experiments for you, and you can rely on their services. So in this slide, it is showing a data that was prepared by an external research group in Russia. For samples that they have was, they got the samples from 18 patients. And for the library preparations, they used the mechanical fragmentation method, and they did the size selections, target size about a 250 base pair. And for the probe capture hybridization part, they followed each product's protocol, and they did a downsampling, which means they reduce the sample read to the 50 million read, so that all samples would have the same number of reads. So it could be a neck-to-neck comparison. So when you look at the data, x-axis is a cover X, meaning it shows the number of the times that the target region has been sequenced, and y-axis is percentage of the target region covered. And so all products showed very high percent target region coverage at a lower read depth, which is a 10x. So specifically, Vazyme, which is a yellow line, Vazyme showed a very high percent target region, which is greater than 98%. So this means more than 98% of the base have been read at least 10 times. And it was the highest above all. And I think this is a great result. And other competitors' products that I've seen before, they don't go higher than 95% of the target region coverage at that amount of the read depth. So this paper showed also other metrics, and overall our product offers performance that is close to that of Agilent and Roche. And this is just a side note, but according to the ACMG, which is American College of Medical Genetics and Genomics, at least 95% of the target region should have equal to or greater than 10x coverage to ensure reliable variant calling. So you can consider screening indels, insertion and deletions, and SND, single nucleotide variants, using our product as well. So, and depends on the sequencing platform type, we have WES reagents that are compatible to each platform, either Illumina or MGI. So for inquiry of specific reagents needed for your sequencing platform types, you may feel free to contact your local sales representatives. And Vazyme provides several panels. On top of the WES panels, we have mitochondrial DNA panels, inherited disease panel, and oncogene plus panel, and lung cancer panels. And all of these panels are based on single-stranded DNA synthesis technologies with high-quality production standards. And it provides a uniform and stable result. And we also support panel customization service to meet personalized demands. So what we provide to you is a one-stop solution. So we can provide the instrument scripts that are specifically designed for your experiments. And we also provide reagents and consumables that are compatible to each other. So we just single call will suffice your need. And we also provide a WES software that meets various analysis requirements in genetic screening and oncology diagnosis. And at this point, it is still under development. And so the software will be launched hopefully soon, and hopefully in the middle of April. But the following are some of the features anticipated. So it is a software that you can actually download and run it. So compared to the cloud-based service, it is fast. And it's a user-friendly interface. And so it is developed to assist in the classification and reporting of alterations from any next generation sequencing platform. And more features will be revealed after launching. So this shows a pipeline of the automated WES solutions. So the blue means the process can be automated. Gray means this process cannot be automated on Vazyme workstations. So we do have experiences of carrying WES customers. So there is a growing number of customers worldwide, including these customers have already benefited from our automated solutions for WES. And more importantly, and they are satisfied with our products and service. So to Vazyme, there is a mature WES series of solutions. It's a hassle-free one-stop solution, including consumables, reagents, and instruments. And several customers from different countries have tested not only our kits, but also instruments and consumables, and they continue to purchase our products. And we provide ready-to-use reagents in plate packaging, and customized panel is also available. And we believe that manually performed experiments are more prone to errors than machines-driven automatic solutions. So why not avoid mistakes and save times? And if you don't have experiences with WES, and we can help you implementing the project, and professional technical team provides timely on-site service, and it'll be cost-effective solutions that can save funds to the greatest extent. So with that, thank you so much for your time. And now I'd like to entertain your questions.